All right. Okay. So I think I'm going to check, make sure everybody's here. Does it look like everybody's here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 2 people are missing. There's a SpongeBob and a SpongeBob background. Excellent. Are you guys all very quiet, or is it just not working?
I think it's not working. We should just call off today.
Yeah, if only. If only. I feel like today I had one of those days where I was like, I'm gonna just record it and send it to them. But I didn't. I didn't. All right, so we have been talking about how to use phylogenetic trees to understand virus evolution mostly, and I want to kind of do at least one more topic today about that before the exam, and then everything after that, whatever we do on Monday won't be on the exam. Okay, so we left off with this guy. I find this cartoon entertaining, like the surprisingly green parasite's funny to me. So we were talking about what coevolution is, and then what it means in terms of the parasites. And so when we look at, we've been looking at phylogenies in this way, and that is to show where viruses come from. And so what we've been showing is mostly that something like COVID-19 has come from a single infection, and that is the only disease that's caused by this particular virus. HIV shows this slightly differently. So you can see on this phylogeny, this is a phylogeny of viruses called simian immunodeficiency viruses that come from primates. And the way that they've labeled these, just so you know, at every tip is an individual virus from one species of primate. And if it says SIV, that's simian, meaning it's from a primate. If it says HIV, that means it's from a human. And what you see is that the arrows point out the places on the tree where HIV shows up. And you can see here we have one arrow indicating HIV2, another arrow down here indicating HIV2, and then at the bottom of the tree, HIV1, and that shows up twice as well. So what this is telling us is not just that the source of our virus is coming from a primate, but it's coming from different primates. So HIV1 is coming from chimpanzees. That's what CPZ is. HIV2 is different though. It's either closely related to what we call the sooty mangabe. That's one of the primates. There are HIV2s that also come from a different group of primates, either macaques or sooty mangabes, depending on how you read this phylogeny. And so what this is saying is basically that these viruses, these zoonotic viruses, can come from multiple hosts. Benji, question.
So some of the I forget what they're called, but the splits have bootstrap numbers and some of them don't.
Mm-hmm.
Why is that?
Usually when people choose to do this to make it easier to read. So if you imagine you could put a label on every single one of those ancestors or nodes,
right?
You get really busy on a tree this big. So they choose to put on the bootstraps at a cutoff, and that cutoff can be arbitrary. Here it looks like it's 75% or better.
Okay.
So that's probably what it is. So if you see something that doesn't have support, like here, I was about to draw anyway. So here, that probably means that we know that this SIV falls into this group. We just don't know how the taxa within it are related.
Okay. Follow-up question. HIV1 is in two separate. Oh wait, never mind. It's different strains of HIV1. Is that what it is?
It means that we know the thing that causes the disease, HIV1,
right,
has come out of chimpanzees multiple times.
Gotcha.
That's exactly what it means, and you see the same thing for HIV2.
Right.
It actually looks more like HIV2 happened 2 or 3 times, but we don't have a full number because the support's not very good, so we just don't know there. okay? So this is
Right.
basically the same thing we've been seeing with SARS, right? It's coming out
Right.
of bats, it's going into humans, but it's coming out with different diseases or different sets of symptoms that humans have, right? And HIV-1 and HIV-2 ultimately cause AIDS, but HIV-2 is much more rapid, causes rapid disease, and it's really, really acute. So it's really virulent. So they are somewhat different. Okay, so that's not all that phylogenies can do with virus evolution, and so I want to talk about some of the other things. This one's nice though because it's pretty and there's a lot of data in it, and oh yeah, I have to turn off the annotation. Clear drawings. And then I should be able to quit that. Go. All right. So what we've been talking about and what we'll talk about again today and try to finish up this idea today is one component of phylogeny. So we talk about the shape of the tree as the phylogeny. And what we mean is that in this phylogeny, you have these 3 taxa, right? And the relationship is such that the top 2 form a clade together and the third is the sister to that. And when you talk about forming a clade and the sister groups to the clade, that's the topology, the shape, the overall shape of the tree. The second component is how long those branches are. And I don't want to get into the branch length stuff before the exam because I think it's complicated. So I want to focus today on topology. Topology tends to answer questions like, where did the pathogen come from? How many times did it show up? Is it co-evolving with the rest of the species that have the pathogen? And then when we get to the branch length stuff, we can start answering questions like, how old is the pathogen? How long has it been evolving with humans? Okay, and so we'll get to it, but just not before the test. All right, so again, we're talking about relationship of who is in a clade with whom. That's really what we're getting at right now. Okay. And all right, and then I probably had to clear that. All right, so one of the things we started talking about at the end of class was this idea of coevolution. And remember, that's when one species evolved in response to changes in another. And when that happens, you can get this representation in phylogenies where the species that are pathogens have the same relationships as the hosts. So in this diagram, it's really a cartoonish diagram of a tick-like or ectoparasite. And then you would see that individuals, this parasite could only infect this host, and this parasite would only infect this host. And these 2 are sister taxa, and these 2 hosts are sister taxa. Okay? In this process where you see that formation of new clades is co-speciation. And you can start to ask questions like, how do you describe the processes that lead to this tree? Well, that would be coevolution. So something like a change in this, I'm going to label them 1 and 2 here. So here's host 1, host 2. So there are unique features of host 1 that allowed it to not be infected by whatever this ancestor was, but along this branch, the parasite evolved to be able to infect host 1. But along this branch, this parasite couldn't infect host 1, it can't do it, but it can infect host 2. Okay, so we can then start asking the questions, well, did this happen within a host or did it happen in the ancestor of the hosts? And so we can start answering these questions. So what's the circle of the title for?
The wording is confusing.
Okay, so what that means is, all right. Is that helpful just to go through all the slides really fast? Okay, so what this means is the numbers are gonna mean that they have a pathogenic relationship with each other. So the tick-like thing infects the one with the number 2. So number 2 infects number 2. And the relationship is that the hosts, these 2 form a clade together, hosts 1 and 2. Ticks 1 and 2 form a clade together in the same topology as 1 and 2, okay? So in a phylogeny, typically when we have this node and then we get 2 species coming off of the tips, this node would represent speciation. So that means that at the same time host 1 and 2 became species, parasites 1 and 2 became species. Does that make sense, Madison? And then you see the same pattern down with the other hosts and ticks where hosts 3, 4, and 5, and ticks 3, 4, and 5. 4 and 5 are sister taxa both in the host and the pathogen, and then the number 3 is sister to 4 and 5. So now they have the exact same phylogenetic relationships in their separate trees. Does that make sense? So that's what we mean when we say co-speciation is represented by the fact that they have the same relationships in the trees. Okay? All right.
Yeah, I think the title's just might be missing a word, and I think that's where she got confused. Like, I feel like it should say when trees show identical relationships.
Oh, maybe it does, and it's just not presenting. Okay, yeah, that's it. Sure, show. 5 years later.
Because I was confused at the same thing. Like, I understand what you're saying.
That's fine. The concept's key there, but 5 years of that title and no one's noticed before. That's awesome. So it turns out that however I formatted these titles, just so you guys know if when I upload the slides and you download them and play with them, there's something about the formatting of the titles to all of these that causes PowerPoint to freeze. So I've tried to fix it, but that may be why it's missing some words. Yes, Jarrett.
So if we were to like continue the phylogeny on,
Yeah.
could we see like a speciation event from species from host number 1, and then like a matching speciation from pathogen number 1?
Yeah, so what you get something that looks like, let me clear out all the existing annotations first. So I think what you're saying is this, that.
So is it always led by the host, like developing some sort of resistance?
Usually yes, because you wouldn't expect the pathogen to cause it to evolve, right? It doesn't want to have that pathogen. So you'd get, this would be 1A here, 1B there, and then this would have, can't draw them perfectly here, 1A and 1B. That's a B. So 1A would infect 1A and 1B would infect 1B. So that's what you're asking, right, Jarrett?
Mm-hmm.
Okay. All right, so when we have really good representations of coevolution, people will actually draw these phylogenies and publish their data showing this reciprocal change in relationships. So when one changes, the other responds, right? But that's not always how we represent this in phylogenetics, especially with pathogens. So when we do this, we can actually look at this a couple of different ways. All right, so to show you what I mean, I have to actually show you guys the phylogeny of primates because I don't have this assumption, this underlying assumption that you guys all know who your close relatives are, because I sure don't know it that well. Well, I do now, but I don't think that I would just say that someone like me who studies frogs is going to just know all the branching points in hominoids. Okay, so hominoid, this term on the right here, refers to all of the apes and then their close relatives. So everybody in the clade that follows here, of course, it's not going to work now. This clade hen, I'm not using annotations right now because it's not working right for me. So this is the hominoids. And just for you guys, for common names over here, the genus Pan includes chimpanzees, pongo, is orangutans, and hylobates are things like gibbons, although there are other stuff in there. I assume you guys know that gorilla, it means gorilla. I didn't think I had to highlight that one. But this also includes within it all of the different hominids, which are us and the Neanderthals and all the other ancient Homo. All right, there's a whole bunch of stuff that goes in there that we normally would have covered at this point, but because we lost a week of class, we haven't done it. So this is us up here, and then the difference is that small clade is called hominids, and these guys are hominoids, so it's different, right? So they're different number of species that are included. Okay, so one of the things that's really important to take from this is that our sister taxon or sister group is the Pan genus. So Homo and Pan form a monophyletic group, and sister to Homo and Pan is gorilla. And so when we're looking at coevolution, what you would then expect to see is that if there's something that Homo sapiens have, some disease, then if it's coevolution, then the sister virus or sister pathogen would be in chimpanzees, and then our virus or our pathogen and the chimp pathogen would be monophyletic, and the sister that should be gorilla. Okay, so we're looking at that topology, that mirroring of topology. So you'd expect to see these same relationships. And so in order to understand if it's coevolutionary, you have to actually know that phylogeny a little bit. So that's why I have this here. Okay. Now, the rest of this tree, it's actually much bigger than this, but it does show monkeys and apes and all sorts of other things. If you're interested in that, I can send you, I think in the comments, the actual link to this paper is in there if you're interested in the phylogeny of primates. But we're going to focus just on what's circled in that pinkish red color right now. Okay, so when you think about what can happen in terms of viruses and other pathogens, what we've been talking about so far because of COVID-19 is there's some evolution of a virus happening inside of a reservoir, and that would be bats for us for COVID-19, and some changes that were acquired in the reservoir are then make it so it can jump into a new host. But there's also this possibility that there's a monkey virus that monkeys carry and had it evolve within them to have different versions of it. So if you were to think about this, each of these little shadows shows one primate with 3 viruses in it. And what it's saying is that this ancestor had 3 viruses, and then the descendant of that also could get infected with those 3 viruses, but they would accumulate changes. Then when it gave rise to humans and chimps, they would have their own versions within each of the descendants of those trees. So what that means, the whole point of even thinking about this, is it means that we can predict using a phylogeny how the virus evolved. So if it was coevolutionary, remember we were looking at those mirrored trees, this is saying something slightly different. This is saying that if it started out in an ancestor, so I'm going to make some labels here to try to help understand this, if I can. There we go. So that's an A if you can't read it.
But,
so what's not shown on here, all right, so what this means is this would be the ancestor, so a hypothetical primate of some sort that carried 3 viruses. Then it diverged into some hypothetical. This isn't a real organism. This is hypothetical. I love writing with a mouse. And then the viruses in here evolved along with the speciation between humans and chimps. Does that make sense to everybody? Yeah, are we following? Okay, so this would mean a strict co-divergence or co-speciation with the host. Whereas in this case, what you're looking at, what you want to point out here is that C1, C2, and C3 all have their most recent common ancestor in humans, right? Whereas here, C1, C2, and C3 are not their most recent common ancestor is way back here, where there's change occurring in different hosts. Does that make sense? Right? So what that tells us is there's predictive ability to go in and say, how do we expect these viruses to evolve? Do we think that we have 3 versions of this one virus because our ancestor did? Or do we have 3 versions because it came from 3 different places at different times? Okay? The key here is recognizing that you can make a prediction by saying, how are these different viruses evolving? And what does the topology of the phylogeny look like? Does that make sense, everybody? So it's a predictive way to do this.
So you're predicting how they will evolve based on how they did evolve? Is that?
Well, so when you make an observation, so I know a lot of you guys took virology, so you guys know things like there are different strains of something like herpes virus, right?
Right.
So there are a lot of them. And the question is, really, it can come down to it's really how long have those viruses been evolving with humans? Is there something about this virus that allows them to make new strains within the human, or have they always been in humans and they're slowly changing with the phylogeny? Does that make sense? It's really a question of, are they evolving, are the new strains evolving within a host, or are the new strains evolving along with the history of the host?
Okay.
So really, when you look at that, you're saying, is the most recent common ancestor of the pathogen, that's what MRCA is, most recent common ancestor, within humans or in another animal?
Okay.
Okay, so you're really looking at this in two different questions. Okay. So the reason I'm showing you this is because we're going to look at a couple trees that show some different patterns. But I want to give you some examples here. So this will kind of take you thinking about what you may have learned in virology. And when you learn virology, you're primarily learning about viruses within humans, correct? Is that correct? You don't really talk about the viruses that have equivalents in other animals?
We do.
We
mentioned
like,
yeah.
But
we did parvo.
Okay.
We
All right,
specifically
so
did a
let's-
lot of the poxes as well.
Mhm.
Yeah. Okay. So when a lot of the poxes, sounds really horrible. Okay. So let's imagine, like I say in this bullet point, that we have 4 different virus variants or strains, if you prefer to call them strains. And you can imagine you could build a phylogeny. If they're really closely related, you can build a phylogeny that looks something like this. And we'll just name them 1, 2, 3, and 4. Right? And that could say, then you know that 1 and 2 share a common ancestor here. 1, 2, and 3 have a common ancestor back here, and 3 is the sister to 1 and 2. And then finally, 4 is the sister group to 1, 2, and 3, right? You'd see that pattern if the pathogen is evolving within the host, right? But it doesn't really give you information without adding into it the context of other organisms, maybe the reservoir, maybe the source of the virus. Okay. So if we add on something like 4 viruses that are equivalent in chimpanzees, and we're thinking about what it would look like on a phylogeny if each of the 4 chimp variants was actually the source of the 4 variants in humans. What you would expect to see is something like this. Maybe, if I can click. Sorry, something scared me about that next slide that's going to show up. So you could have, well, let me go back 1 slide for a second. Right, okay, so this is where we want to be. We don't want to jump into the host yet. Let's just imagine we have 2 separate viruses, 1 in humans, 1 in chimps, each with 4 variants. Oh my God, why is it doing this to me? That's where we started. And we're going here. All right. What's funny here is that I've got to do this with 3 different types of ways to draw. This is what's fun. Okay, so this is human. And then we're going to do the same thing for chimps, and just imagine that there are 4 chimps as well. Okay, right? So that's good. If you're a perfectionist and you like really straight lines, don't ever draw with the mouse in PowerPoint. Okay, so if the viruses are similar, it can't do it on my computer. It's like a desktop computer, I can't do that. So if this is the phylogeny, we just have 2 separate ones, great. But you might ask the question, well, if these are the same viruses, if these are, let's say, pox viruses, there are really different ways that these 2 phylogenies could be related. So if you remember, when we talk about viruses and we give them the same name, they usually have the same gene order, the same gene content. So that means you can build a phylogeny from human viruses and chimp viruses and put them into one phylogeny together. So you could imagine a scenario where it actually looks something like this, where the human viruses form a clade like this, right? That's the human viruses. And then you could have the human viruses, or the chimp viruses form a clade, right? And that would mean something like here, all the human viruses share a common ancestor with each other. Here, all the chimps share a common ancestor with each other. And maybe this common ancestor also had 1, 2, 3, 4, and they speciated together. Does that make sense? Viruses and humans speciated at the same time. That make sense, everybody? Great. So now we're going to go to the slide where we talk about the reservoirs. So really want this to work right. So this was our original phylogeny at the top, showing the just the relationships among human viruses. Now I'm going to imagine a scenario where the pathogen evolved within a reservoir and then jumped to a new host 4 times. And let's call the chimpanzee our reservoir. What you would get in a phylogeny for that would look something like this. And I did practice this before doing this so I wouldn't screw it up. So I don't expect you to be able to do this very quickly because it took me a few minutes. That's at the bottom of the slide. Excellent. So what you would end up with in this phylogeny would be C1, human 1, chimp 2, human 2, oops, human 2, chimp 3, human 3, chimp 4, human 4. Okay, does that make sense, everybody? So what you're saying is then that this virus jumped into humans here. I can't erase with this type of annotation, so I apologize. C2 jumped into humans, C3 jumped into humans, then C4 jumped into humans. Okay. That's pretty cool, right? So the reason I did this sort of hypothetical or this example is that I first show you that co-speciation phylogeny where you lay out the host phylogeny next to the pathogen phylogeny, and no one does that. No one does that anymore because there's this assumption that you know the host phylogeny. So what they instead do is, haha, all right. So what they do instead is they put them all together kind of like we see here. So there, okay. So now when you look at, this is a phylogeny of herpes viruses. If you do not know this, there are herpes viruses in almost all mammals, even in some reptiles and some birds. And if you take and you get the viruses from all of these guys, what you see is that in every case, you see something like this where the virus, so if you look down where it says gamma 1 down on the side, you see that HHV4, that's the human herpes virus, is sister to something within primates, and that repeats itself throughout the tree most of the time. So it's down here at the bottom in HHV8. It shows up with HHV5, HHV3, and HHV1 and 2. Okay? So you see that repeated pattern where it looks like herpes virus is co-evolving with its host. So that means back here, oh yeah, I'm not annotating, hold on. That means back here, oh my God, shit. All of these probably had herpes virus 3 and herpes virus 1 and maybe 2, okay? Does that make sense? And then you could make that prediction throughout. So Alex, I think a while ago, I think it was you who asked about what do the alphas, betas, and gammas mean next to things. This is a really nice phylogeny showing how we do the taxonomy of viruses, because we name them alpha 1 based on the fact that they share a common ancestor back here, alpha 2 back here, and then all of alpha back here. And it's really based on all the taxa that have it and have similar sequences. Okay, so that's why it shows up.
So it's kind of like just different clades, I guess.
It really is different clades. It used to be a lot based on presence of genes and proteins that before we could do the phylogenetics in the same depth we can do it now. This phylogeny is pretty old. It's from the chapter, one of the chapters I've posted on Blackboard. And it's I think early 90s phylogeny of this, if not earlier. Okay, so it's pretty old. Yeah, Madison.
Why are like the turtles and the fowl not included? And like mouse and rat and stuff, they're kind of left out of these different groups?
My guess is that they belong to a different group of beta viruses and alpha viruses. There's two possibilities here. One is that this is, so fowl kind of fall into two different clades. And it could be that there's like an alpha 3 here, an alpha 4, and an alpha 5. But what the book had highlighted is only the stuff that had human equivalents. And so that's my guess at the best of it. I don't know all the details about the herpes viruses in birds and reptiles. So I just don't know. Okay.
Alrighty.
All right, so that's kind of cool. This is one that in the chapter that I posted on Blackboard about, I think it's, well, I'll just look at the title while I've got it in front of me. It's chapter 13 in Stearns and Coella, I think is what I've titled it up there. And it's about evolutionary diversity of viruses. So that one's in there and you have all the reading about it. And so what they've inferred from this is that the herpes viruses, we don't have many herpes viruses because it's evolving so rapidly, we're getting new ones. Instead, we have all these different versions because they are tied to our common ancestor, right? So there were, let's say, 7 versions in our ancestor of vertebrates. And Now there are still 7 versions of that. You know. I suspect trouble is about to happen. You'll see why in just a second. Flying by. Okay. You know it's bad because he's trying to jump on like 7 things and you know something's coming down in a minute. Okay, so it's not just herpes viruses that do this. There's a recent paper out that shows this also happens in papillomaviruses. So papillomaviruses are the ones that cause HPV. And there are a lot of different ones. And this phylogeny, so this one's a little bit harder to read because there's so much information in it. And we'll come back to it when we talk about the role of the branch lengths. But they have in it PP up here. That's your chimps. The MM is a macaque. And then and then there are a couple other things in there that if you're interested in it. So that area you've put your question mark on, I assume that's you, Madison, because you like the question mark. It's showing you that there's a lot more. So if you look really carefully at this, there's a node back here. And I'm going to highlight it with this pen because that seems to be consistently working. So this one, it goes like this and it makes a triangle right in here.
Yeah,
back to
there's a
this.
bunch of them.
So normally what people do when they're looking at a huge phylogeny is they only show the branches of what they're interested in. So what the little triangle would mean, this shape, at the end of a branch is that there's a bunch of viruses in there they're not showing you. But there's a bunch of viruses and this is still a monophyletic group because it has a node. It's just not showing you everything that's in that clade.
Okay, because there was like a bunch of them and I've never seen
there's
that.
a bunch, and I don't know why they did it. To be honest, I didn't fully read this paper. I just wanted to give you guys this because it's cool that it's not just herpes viruses, but it's also papillomaviruses, and that's another one that we have quite a few versions of. Okay. Anyway, so that's cool. And then it seems like there's some papillomaviruses that we're really comfortable with knowing that they came from our primate ancestors. But if you notice down at the bottom, we also don't know where all those came from. They don't seem to fall in the same relationships as others. So that's kind of cool. And that we just don't, it's probably a sampling error. We don't know because we haven't sampled the right thing yet. Okay, so that's cool. All right, so this comes down to the question next of, that's great when they all co-occur and they do what they predict. When a virus doesn't correspond with the evolutionary history, something like what we see with COVID-19 is that the source of the virus isn't from a chimp, it's from another host. And we call that jump from one host to another host switching. Okay, that's kind of a generalized term for the evolutionary phenomenon. And so in this case, we're looking at, it's the simplest phylogeny I could find of host switching, and it's in Drosophila. So Drosophila have their own viruses. So you see the Drosophila phylogeny on the right, and the names of the Drosophila are right here. And you can see that some of these viruses correspond to the phylogeny, but some of them do not. So you can see that the virus that occurs in D. immigrans seems to have jumped from a different virus that it's not most closely related to its host. Okay, so these diagrams are called tangleograms sometimes, and what it looks like in humans is many viruses, when they do a lot of host switching, the number of lines that cross against the phylogeny are so huge you can't really read them. So that's why I didn't pick one of the human ones, but I might be able to pull one just to show you how complicated it can be. So what this is telling you is that these viruses are making jumps. They're not co-evolving with their host. They're making some massive change to the viral structure to allow them to enter something that they're not closely related to. Okay, does that make sense to everybody? Okay, all right. So it's fun to see all the different pictures today because it's weird. Because I love, Isaiah, your picture just makes it look like you're confused the whole time.
I mean,
It's great. Okay, so one of the things Benji asked about the whole idea of how do we get this host switching? How does it happen? And the best data we have is from influenza. And so one of the things we can look at right now, you guys could do it, anybody can do it, is the database of influenza viruses. And so every year, people who study influenza document this, they put it on a database online, and you can do things like make a phylogeny of how it's changing in the current year. You can look at different versions of the phylogeny. And so it's just an incredible database of symptoms, of predictability, and all that. But when you look at the different phylogenies of influenza, there are really two different shapes you get. So this is a really good example of topologies being a little bit different. So on the left side is a set of versions of influenza found in birds. And they're named based on two proteins that are on their surface, H and N. And H and N, they're surface proteins. I'll show you those in just a second, but you can see on the picture in panel A, oops, sorry, there you go. All right, H1N1 in human, H1N1 in swine, H1N6, H2N2, H2H9. And so that's how we talk about them. That's how we talk about different influenza viruses. And you can see that this phylogeny kind of looks more like what we just looked at with herpes virus, where you have different hosts shown there, but all of these samples were taken from birds. So these are influenza viruses that are found in birds. And it doesn't mean they're disease causing in birds, it just means they're found in birds. On the right in panel B is an example of the influenza viruses that you can find during an infection or set of infections. So this is influenza A. Stupid, they should have put that in panel A. So this is influenza A from the years 1968 to 2006. And you can tell the different years based on the shape and the shading of the shape at the tip. And when you look at these two different phylogenies, both of them have branch lengths in them, right? You can see that they're different in length, right? They're not all just straight at the edge, but they're very different. And on the right, we call this a ladder-like topology. So it's almost like you're climbing a ladder. And you can see that there are slight changes in the nucleotide sequences as the years progress. And you're not going to find something from 1968 in the flu viruses from 2006, right? So that shape goes away after each set of years. Okay, does that make sense, everybody? Okay, and so the point of this right here is just to say that you can get two different shapes of a phylogeny. Okay, Isaiah, what's your question?
Yeah, so there's this thing that happens with flu. It's the antigen drift versus antigen shift. And
Yeah,
so
we're
I'm
gonna get
wondering
to that.
how, okay, I was about to,
Go ahead.
like, how does it handle difference between like explicit mutations versus complete swapping of,
Well, you said, how does it handle it? What is it?
like like someone who does phylogenetics? Like, can you represent both in the same phylogeny, or do you have to approach it from?
So what you're looking at in the left is hemagglutinin, so the H sequences for several different bird viruses. All right, so I think that the answer to your question is this is how we deal with it. So on the left side, all of the sequences are from when it says H1N1, all you're looking at there is the sequence, the DNA sequence, RNA sequence from the H.
Oh, okay.
So you might do like some diagnostic of this particular flu. It has both H1 and N1, but then you only take out the DNA sequence or the RNA sequence for H1.
So in reality, it's probably a slew of different like identities within the H1N1, but it's just simplified here? Or is that, because like even within H1N1 human, I'm sure that there is, I don't really know what I'm trying to ask here.
So basically, so just think about H1N1 for a human for a second, okay? H1N1 in a human, if we have immunity to it, should look like what's in panel B over time, all right? Because the reason that you're getting that ladder shape is the virus is undergoing small enough mutations so it can evade the human immune system, right? That's what's causing this change. So if the human immune system can respond to H1N1, you would over years see this same ladder-like phylogeny coming off of the tip that you see right here.
Okay, that makes sense.
Okay, and then you should see the same thing in swine and the same thing in H1 plus N6, etc. Okay.
Okay.
But you're asking, the question that you're having trouble asking is a little bit more difficult to answer with the slides I currently have. So and this is partly because of, it's partly because of the limitations to data when these original figures were created. Okay, so let me on Monday show you a full genome of influenza viruses, because I think that'll help answer your question a little bit better. Because right now we're only looking at one gene, so it's only, this is really only showing you the H virus phylogeny. Okay? Does that make sense?
Yes, ma'am, it does.
Okay. All right. So Isaiah just brought up this idea that there are two types of evolutionary change that happen in influenza. And so what we see in panel B is something called antigenic drift. So an antigen is just the surface proteins that our immune system responds to. And so in the case of antigenic drift, what's happening is the surface of the virus has these purple circles. If they make small changes in their shape by changing their nucleotide sequence, then you get a different shape, and our immune system has to go through the process again to recognize that shape to fight it off. And so every year, every time the flu comes around, we have to develop new resistance or new response to a new shape of the flu. Antigenic shift is wildly different, okay? So antigenic shift means it's not just these small mutations in the viral sequences that normally infect humans. It's like one virus, the purple one that normally infects humans, exchanges pieces with two different other viruses to make a brand new combo of viruses. All right, so it's actually making huge changes to the outer surface of the virus. All right, that is called antigenic shift. People think it's fun to rhyme things when all of us who teach are like, that's even harder. Yeah, Madison.
Okay, so like, I didn't realize the microphone was right there, so if you heard me breathing, I'm sorry.
I
But
didn't hear it, so it's good.
so for antigenic shift, like they both have to be in the same host at the same time, at least for influenza. When you do it on a phylogeny, would you then have it converge from separate ones?
It depends. I
Further
think
down?
that's also what Isaiah's asking. So I'm going to show you a full genome Monday, but what I want to show you, what we were looking at in the left side of that last slide is just one of the proteins on the outside of the flu virus building that phylogeny. Okay, so
Yeah, because I know that H1N1, like H is one factor, but I think N is the more virulent factor, so they make
it is.
different.
So when you look at, so here's what it, so when you take a virus sample, when a virologist takes a sample, they get what's called a serotype, and that characterizes what type of hemagglutinin you have and which type of neuraminidase you have. So those both are proteins on the outside of the flu virus. And what happens is when you get an antigenic shift, you change one of those to something that's not typically in humans. And so when you look at the phylogeny here, what you're seeing is each of the clades is really H1, right? So they're based on the H's. So each clade is one version of H. There's 1, there's 2, there's 5, there's 6, there's 8, there's 12, and 9. And you'll see that the H's all form a monophyletic group. You could do the same thing with N's and you'd get a totally different topology. You'd get the N1s grouping together, the N2s grouping together, the N6s, okay? But what happens is then you get conflict in the phylogeny. You would get those crossings between, oh, wait a minute, this looks like a bird flu versus a human flu, okay? Okay, so when we talk about this, so Madison just brought an interesting point up to say that in order for flu viruses to do this, they have to actually be somewhere where all the viruses are in contact with another. And what we were talking about with the COVID-19 and HCOV2, yeah, Benji, do you have to go?
Yes, ma'am.
Okay, is that your question or did you have to do something else?
No, that was my question.
Let me just do one quick thing here with flus because I think,
Okay, sure.
so the thing about flu that's weird is that it doesn't have one nucleotide sequence, it has 8.
Right.
So when it uncoats, what happens is it dumps off all of these nucleotides into a cell, and then when an envelope reforms after replication, you can gather up all those different versions together. So it's not really like recombination like we think of with crossing over. It's actually just a reassortment. It's like the envelope collects the wrong, an envelope isn't specific about which ones it's going to collect. Okay, so this is one way we can recombine viruses to get brand new strains of viruses. Okay, does that make sense? And I'll stop at reassortment, but this will get at, again, other ways that viruses can do this, because this is not how SARS is doing it, right? It's not how SARS is doing it. Okay, I'm gonna stop there. Are there any questions or anything like that?
Is that where the material for the test ends?
Yes, it is. Absolutely, right there.